Hello. Today I will be presenting Scanning Electron Microscope Validation Analysis by Adan Jose Esquivel Jr. Scanning electron microscope with energy dispersive X-ray or SEM-EDX. SEM-EDX is a powerful tool used in various fields of study and quantify the elemental composition of an observed specimen. Characteristics of SEM-EDX is to detect nanoparticles and is considered in use of all works that require elemental determination, either endogenous or exogenous factors within cells, tissues, and other samples. Knowing SEM-EDS or EDX function and capabilities. SEM-EDS, or more commonly known as EDX, is a combination of a scanning electron microscope and energy dispersive X-ray to analyze samples based on the generation of characteristic X-rays in the atoms of the specimen by incident beam electrons. The scanning electron microscope aspect provides the imaging, and the energy dispersive X-ray is used for the detection and analysis. Scanning electron microscope functions by generating a beam of electrons from an emitter cathode within the electron gun component. The beam is accelerated and focused by the anode with a series of electromagnetic lenses. This allows for scanning of the sample surfaces and interacting with the molecules of the sample, emitting secondary electrons from the surface. Energy dispersion X-ray spectroscopy. The emission of the electrons are then detected by the EDX detector, measuring the energy of the X-rays emitted from the electron beam when hitting the sample surface. With this, the electron beam provides information on chemical composition and knowledge of internal processes within a sample. SEM-EDX uses in microscopic and elemental analysis. The range of SEM-EDX in microanalysis is considered to have high sensitivity in detecting and providing semi-qualitative and quantitative results on various specimens. To obtain microanalytical results with EDX requires for specimens to undergo dehydration to preserve the elements present in their physiological active site, following with incubation in 95% ethanol, absolute ethanol, and propylene oxide, leading to the process of the specimen embedded in plastic to secure the structure for analysis. As for ways to detect elements for SEM-EDX is to detect the impact of atoms with electrons that generates two fundamental physical events called elastic scattering that decides the shape of the interaction volume, and inelastic scattering responsible for the size of the interaction volume. Elastic scattering involves a change in direction of the electrons with no energy loss caused by interactions with the nucleus comprising the materials. For inelastic scattering is the loss of energy with no change in direction of the electrons caused by interaction with both the bound electrons and nucleus in the atoms. Identifying microscopic and elemental analysis in the journal article. In the journal article I chose involves the use of SEM-EDX for urine analysis, a powerful tool to further have in-depth details on ultrastructure and elemental composition of a specimen. This investigation uses SEM-EDX as a microscopic analysis on urine sediments to identify different urine components on a total of 206 urine samples with or without urinary tract infections present. Identifying in the urine sediment samples such as microorganisms, crystals, epithelial cells, leukocytes, erythrocytes were focused when analyzing the urine sediment samples based on morphology, size, contrast, and elemental composition into classifying categories below. Microorganisms in urine sediments, categories that include gram-negative bacilli, cluster cocci, chain cocci, gram-positive bacilli, and yeast. Epithelial cells and urine sediments into categories of the types of epithelial cells like renal, transitional, and squamous epithelial cells. Leukocytes and erythrocytes are detected with various morphological forms such as dysmorphic and isomorphic. SEM-EDX different sampling methods. With the probability sampling method includes the entire population, which in turn offers an opportunity to being selected at random, mainly utilized in quantitative research, and the results produced are more valid representation of the entire population. Probability sampling involves random selection and allows for strong statistical interference about the whole group. Simple random sampling, every member of a population has an equal chance of being selected. Systematic sampling, similar to simple random sampling, but listing the population with a number instead of randomly generated numbers. Stratified sampling involves dividing the population in further subpopulations based on characteristics. Cluster sampling involves the dividing of populations into subgroups and randomly selecting the entire subgroup from the population. SEM-EDX sampling method in the journal article. The study is a sampling method of a random sampling for SEM-EDX to analyze 206 urine sediment components at random with identifying based on concentration, morphological, and chemical composition, regardless the presence of urinary tract infection or not. Having the SEM-EDX blindly at random perform crystal measurements and identification with the use of a detector. Dividing the urine samples into two, one half examined through standard routine urinalysis method with the use of the UF4000 urine culture analyzer. The other half used an SEM-EDX analysis requiring the sample preparation to be completed between 1 hour to 4, beginning with centrifuging 2 ml of the urine specimens at 8,000 RPM for 5 minutes, following with obtaining only 100 of the microliters of the pellet into the sterile slide for a cytocentrifuge machine at 800 RPM for 7 minutes prior to analysis by SEM-EDX. Evaluating the conclusion obtained by the author of the journal article. By identifying the strategy of the researcher in the article study is by recognizing features such as shape, arrangement, border, presence or absence of spores, and other genus-specific characteristics. Overcoming obstacles like bacteria with similar morphology and cell wall contrast by instead measuring and differentiating the bacteria with. As a final identification in the article study is results are compared to the outcome of gram staining carried out with the use of a Sysmex UF4000. This confirms the results of both the SEM and gram staining are identical, exempting the polymicrobial sample in SEM if at least one of the bacteria present is matched to the gram staining. Conclusion: in what ways could the evidence evaluation or validation study should have improved or praised on? With the use of SEM-EDX for crystal identification in urine samples, 83 out of the 206 total urine samples had a presence of crystals, specifically calcium oxalate being the majority, following with styruvate, also known as magnesium ammonium phosphate, second most common. The SEM-EDX provided rapid and accurate results for the classification of microorganisms that were present in the urine samples, such as gram-negative bacilli, gram-positive bacilli, chain coochie, cluster coochie, and yeast. I praise a wide range of viewing samples with the SEM, making it possible to identify details on general description of urine composition, as well as preliminary identification of the microorganisms and cells. While EDX analysis provides elemental chemical composition of the crystals present in the urine samples, allowing for more specific identification, this strategy of combination of SEM-EDX coupled with flow cytometry proved that effective and should have further studies in validating this collaboration for urine sediment analysis in clinical laboratory practices. These are my references. Thank you and have a great day.